regards salivary glands, we have two types of salivary glands, minor and the major salivary glands. Where we can find the minor salivary glands, they are present in submucosa of tongue, cheeks, and belly. What? Turn it off. ده اللي يعجبه.
كده تمام.
Okay. So minor salivary glands can be present in legs, cheeks, tongue, palate, and so on in the submucosa. They are simple or single branched glands. Mainly mucus, but sometimes may be mixed. As regards the major salivary glands, we have three types of major salivary glands. We have two parotid, two submandibular, and two sublingual. Each one is formed of stroma and parenchyma. What is the stroma? Is the connective tissue. The parenchyma, secretory units, and the duct system. The stroma in the form of capsule, septa, and inside the lobules, connective tissue in between secretory units and the duct. What is about the secretory units? Secretory units may be serous acini, which form motor secretion rich in enzymes. Another type, mucus acini, which secretes mucus. The third type, mixed acini, which are formed of mucus acini with a crescent of gynosis. The crescent is formed of serous cells. So it contains mucus acinus with a crescent of serous cells. Who sweep the secretion of mucus acini and the serous acini? Myoepithelial cells. What about the duct system? The duct system inside the lobule is formed of two types. Number one, intercalated. The intercalated duct not apparent in action. The intercalated duct takes the secretion from the mucus and serous acini and deliver the secretion to striated duct. This is striated duct. The intercalated is lined by lobular epithelium. The striated duct has basal striation. They modify the secretion of the salivary glands and also secrete an enzyme, lysozyme and lactoferrin. Also transports antibodies formed by plasma cells in the connective tissue into the saliva. Okay? The secretion is carried from intralobular duct to interlobular duct to the main duct, which open into the mouth cavity. Okay? The parotid gland is pure serous. It is formed only of serous acini. And it is characterized by the presence of fat in its receptor. So, fat is characteristic to the parotid, together with the presence of only one type of acini, which is the serous acini. One important thing that the tissue then passes through the parotid gland. As regards the submandibular, it's mixed but mainly serous. So it contains many serous acini than mucus acini. As regards sublingual, it's mixed but mainly mucus. So we discussed the salivary gland, or finished the salivary gland. Okay. These are mucus. This is sublingual salivary gland. These are striated duct, which modifies the secretion. They are intralobular, and these are intercalated ducts. The small ducts lined by cubical cells. This is sublingual. sublingual 2 or mucus sinai and enter back down. Forget about the tongue. What is about the tongue? The tongue is a mass of skeletal muscles connected by connective tissue. It is covered by a mucous membrane from outside. As regards the muscles of the tongue, it is present in three main directions. What are these three main directions? Longitudinal, from posterior to anterior, or anterior to posterior. Vertical, going from down to the surface, or transverse. So the skeletal muscle, the tongue contains skeletal muscle, present in three directions, to make the tongue able to move in different directions. Okay, what's about the mucous membrane? The mucous membrane is covered by stratified squamous epithelium, but at the surface. The dorsal surface of the tongue is divided into anterior two-thirds and posterior third by a groove called sulcus terminalis. What is present on the dorsal surface of anterior two-thirds? Projections. These projections are called papillae. Okay? There are mainly three types of papillae in human, and one is rudimentary. What are the three types of papillae present? This is one type. The most common type is the filiform papillae. They have a broad base and a narrow base. They are characterized by two things: covered by stratified squamous keratinoid, but no taste buds. What is about the fungiform papillae? Okay. And this is filiform papillae. These are filiform papillae. And this papilla is the fungiform. How from the base and distended from the apex, it has its taste buds are present on the apical surface. They are present at the anterior part, the tip of the tongue, and the side of the tongue. Number 3, circumvallate papillae. By circumvallate, they have a surrounding groove. See, surrounding groove. Okay. It is mostly under the surface, but just projecting above the surface. Under the surface and surrounded by a groove. They have taste buds. Their taste buds are present on the side. The groove is cleaned by the secretion of serous glands. These serous glands are called von Ebner's glands. About foliate papillae, they are rudimentary in the tongue. The posterior third of the tongue contains the lingual tonsils. Yes. Because each one is formed of... No problem. Okay. This is the taste bud present in the tongue. It is formed of three main types. Many types, but three. Number 1, in the center and the base sensory, the surrounding are supporting at the base is stem cell, which divide to replace the lost sensory or supporting cell. Taste buds, the tooth. The tooth or the teeth, its main mass is formed of the dentine. Each tooth has a crown projecting above the gum. Each crown is formed of dentine covered by enamel. In the root, the dentine is covered by cementum. The tooth has in the center a bulby cavity forming of loose connective tissue containing the blood vessels and nerve fibers. What is about the lining of the bulby cavity? The bulb cavity is lined by cells called odontoblasts. These cells are responsible for the formation of dentine. The odontoblasts or the dentine contain cavities, tubular cavities. These tubular cavities contain tissue fluid together with processes of odontoblasts which reach to the area of junction between enamel and the cementum, dentine and enamel, and the dentine and cementum. It also contains tissue fluid, and in the proximal part it contains nerve fibers. If tooth or teeth caries, the Swiss, reach to the dentine, infection can go through and stimulate the nerve fibers. What is about the mineral content? The mineral content is the highest in the element. It forms about 95 or 96 or a little bit more. In the dentine, it's about 75. In the cementum, 65. Okay? The cementum is not cellular in the upper part, but cellular in the lower part. Who connect the tooth to the alveolar bone? Periodontal ligament. The periodontal ligament gives the blood supply to the cementum, and it contains the receptors for which detect the pressure on the this is quite sufficient for the okay, the hard palate or the palate. The palate is formed of anterior bony part and the posterior movable or non-bony part. The bony part is covered by the mucous membrane. The mucous membrane is stratified squamous keratinized. In the submucosa, you will see glands. In S is the submucosa. It contains minor salivary glands. The mucous membrane is attached to the periosteum of the bone. The hard palate separates the mouth cavity from the nasal cavity. The soft palate is formed of muscle. In the center, the muscle is covered by the mucous membrane. Mucous membrane at the side of mouth cavity is stratified squamous non-keratinized. In the part towards the nose, it is covered by respiratory epithelium. During the swallowing, it goes up to close the posterior opening of the nose, preventing food from entering the nasal cavity. Left. The left is a mass of skeletal muscles. Its direction is circular, orbicularis oris. The mass of muscle which runs circularly in the left is covered from outside by skin, thin skin. It is covered from inside towards the nose cavity by the mucous membrane. But the type of epithelium here is keratinized. And the skin is provided with hair follicles together with a sweat and sebaceous gland. Because the covering epithelium is stratified squamous non-keratinized. In the submucosa, mucosal gland, labial mucosal gland, which represents one side of the minor salivary gland. As regards the red mark, which is present in between the skin and the mucous membrane covering the lip from inside. It is covered by modified skin because it's present in the surface, so nothing other than the skin covering the body. This skin is modified because it doesn't contain hair follicles, and mostly and usually does not contain glands. The upper lip may be, but forget about it. Who gives the color for the red margin? The rich blood supply. Where you can find the rich blood vessels in the left? Suppose this is the red margin. The underlying connective tissue will go to the epithelium. So this is the epithelium and this is the connective tissue. So these are called dermal nervity projections containing or rich in blood vessels. And it is also contain receptors for touch. That's it for the leg. What about the cheeks? Okay. The iris is covered posteriorly by 2 layers of pigmented epithelium, while anteriorly by connective tissue cells and melanocytes. Is it vascular? Yes. Does it contain melanocytes? Yes. Do the iris contain 2 types of muscles? Yes. What are the 2 types of muscles in the iris? 2 types of muscles. And third is the pupil constrictor pupilli muscle. Third is the posterior surface, the red area here is formed of dilator pupilli muscle. Okay? What is the nature of constrictor pupilli is smooth muscle. What is the nature of dilator pupilli is what? Okay. This is the constrictor pupilli and this is the dilator pupilli. The dilator pupilli is formed of processes of the pigmented cells containing actin and myosin. They are arranged parallel to the posterior surface. They can pull on the pupil, dilating the pupil. So they are called dilator pupilli. They are processes from the inner layer of pigmented, from the outer layer of pigmented epithelium of the retina. While constrictor pupilli is formed of smooth muscle. Okay. What is about the lens? What is the structure of the lens? Do the lens is surrounded by a lens capsule?
Yes.
Yes. Do the lens have an epithelium?
Yes.
Where you can find the epithelium in the lens? Supracapsular or subcapsular? Subcapsular. What is the function of lens epithelium? What is present inside the lens? Lens fibers. Who forms the lens fibers? The subcapsular epithelium. At what site in the lens? The equator. Do the process of transformation or formation of lens fibers is continuous all over the edge with age? Yes, continuous. So the lens becomes larger, or the lens, because it contains crowded fibers, its elasticity decreases. And its elasticity is important for accumulation. Okay. What else? Do the lens fibers contain a protein, a specific protein?
Yes.
Crystalline. Protein called crystalline. Who pull on the lens? Zonule fibers, which are attached from one side to the ciliary body and from the other side to the lens capsule. Who, with contraction of muscles in the ciliary body, the relaxation of the new fibers occur, and the length becomes thicker. With relaxation of the muscle, the new fibers are distended, and the length becomes thinner. Now, what is the structure of ciliary body, which is a part of the uveal tract? The uveal tract is the middle tract, which is formed of vascular, pigmented, connective tissue, formed of iron anteriorly, ciliary body, and then the choroid. So, what's the structure of ciliary body? Vascular, pigmented, connective tissue, containing smooth muscle. What is the name of this muscle? What is the type of this muscle? Smooth muscle. Present in how many directions? Three directions. Okay? Radial, meridional, and circular muscle. What is the other function of ciliary body? Secretion. Secretion of what? Aqueous humor. What are the types of cells? How many layers of cells covering the ciliary body and the ciliary processes? Two. Do the two are pigmented? No. The inner one, that is the muscle, is pigmented. While the other one bordered, the inner side of the eye is non-pigmented. Okay? And in between the ciliary processes, the new fibers are attached to the ciliary body. Okay. Retina. This is the sclera. What is the type of tissue? Type of tissue formed of what? Regular or irregular? Regular. Dense? Connective tissue. Next to the sclera, choroid, which is vascular, pigmented, connective tissue. And then next is the retina. What are the layers of the retina? 10 layers. Number 1 from outside, pigmented epithelium. Next, rods and cones. Next, outer limiting membrane. What is the nature of outer limiting membrane? Site of cell junction. Cell junction in between the supporting cells and the rod and the cone cell. What is the supporting cell? The molar cell. Where you can find the nucleus of molar cell? In the inner nuclear layer. Okay. This is the area of rod and cone. This is the area of outer limiting membrane. This is the area of outer nuclear layer. What does the outer nuclear layer contain? Cell body and the nuclei of rods and cones. Next, the outer plexiform layer, site of synapse, in between what? Rod and cone cells and bipolar nerve cells. What are the types of cells or the nuclei of what types of cells are present in inner nuclear layer? Bipolar, amacrine, horizontal, and molar cells. The next layer is inner plexiform. Synapse in between bipolar and ganglion cells. Next to the ganglion cell, what is the origin of optic nerve fiber? It is the axon of which type of cell? Ganglion cell. Is it myelinated or unmyelinated? Unmyelinated inside the eye and become myelinated outside the optic disc. Next, nerve fiber layer, after the ganglion cell layer, nerve fiber layer, and then the inner limiting membrane. Is there an area only contains cones?
Yes.
Which area?
Yes.
What is the character of cones present in fovea centralis?
Long cone.
Long and thick. Long and thick. Longer and thinner. Do the fovea is vascular? Avascular. Avascular. Does it contain rods? No rods. Okay, so the optic disc is the blind area, while the fovea centralis is the point of maximal visual acuity. Sensitivity. Okay.
Cochlea.
Okay. What are the contents of the middle ear? What are the contents in the middle ear? Does it contain bone? Yes. Middle ear or inner ear?
Middle ear.
What are the contents of the middle ear? Does it contain bones?
Yes.
What are the names for these bones? Malleus, incus, and stapes. Which of them is attached to the primary tympanic membrane?
Stapes.
Malleus.
Malleus.
Malleus. Which of them closes the oval window? Foot of the stapes. Okay? Where you can find the secondary tympanic membrane? Closing what?
Oval window.
The round window. Who closes the oval window? It actually acts as a bump. Okay? What is the lining epithelium for the middle ear cavity? Simple squamous or low cubical epithelium. Connected anteriorly with the pharynx through Eustachian tube and connected posteriorly with the mastoid air cell. Okay. What does the vestibule contain? The inner ear is formed of membranous labyrinths present inside bony labyrinths and separated from each other by space containing perilimb. The wall of membranous structures are formed of connective tissue covered by simple squamous cells. Okay. What are the membranous structures present in the vestibule? Utricle and the saccule. What is present inside the cochlea? The cochlear duct. What is present inside each semicircular canal? Semicircular duct. The space in between the sacs and the wall, or the duct and the wall, contain perilimb. While inside the sacs or the duct, endolimb. Do we have receptors in the inner ear?
Yes.
What are the receptors present in the inner ear? Receptors for hearing, called organ cortex. Receptors of equilibrium present in utricle, macula, utricular. Saccule, macula, saccular. It's an ampulla of each semicircular duct. Cresta ambularis. What is the general structure for the receptors in the inner ear? Three structures: hair cells, supporting cells, and gelatinous material. Okay? What is the difference between hair cells in the organ of Corti and the macula in utricle and saccule? Guys, what else? Do the gelatinous material contain something? Yes. What is this something?
Crystals. Crystals.
Calcified.
Calcified.
Autolysis.
Autolysis, yeah.
Autolysis. Macula utriculi and the macula sacculi. Its gelatinous material is characterized is called autolytic membrane because it contains autolysis, which are calcium carbonate crystals. Source of calcium carbonate crystals. What is about the gelatinous material in the cresta ambularis? Cone-like or elongated without autolysis. Okay. Now to the organ. What is this? What is the structure number one?
Scala vestibuli. Stibuli.
Number two.
Scala tympani.
This is structure. Organ of
Corti.
Number four.
Vestibular membrane.
Number five.
Ligaments.
What are the limits of the cochlear duct? The boundary of the cochlear duct is formed of what? Scala. Scala media. Vestibular membrane. From down?
Basal membrane.
From lateral? Okay. What is the structure present inside the modula? Groups of cells, what they are called? Spiral ganglion. It's a membrane code to receive the impulse from hair cells to the cell body and send it through axon, which is the cochlear nerve with the vestibulocochlear. It's the cochlear division of the vestibulocochlear nerve. Now, number 4, vestibular membrane. Number 11, basilar membrane. What is this study? What is this study? What limits or supports the wall of tunnel cords? Who supports the hair cells? Pharyngeal cells. Okay. Is there a difference between the basilar membrane at the base of the cochlea and the basilar membrane at the apex of the cochlea? What is the difference? So, the two sides are different in stimulation. The loud sounds affect the basal of the apical. The basal. While the low-pitched sounds affect so the lower is broader. The lower, its fibers are thicker, while the apical is less in the breadth. Its fibers are thinner. Okay, and there is a difference inside of stimulation. One second. I finished.
Thank you, doctor.
Do you have a question?